Welcome to Biosport for You, the channel for discussions in biology. I am Sijo Matthew. With me,
Prasanna Rajan. In this video, we are going to discuss about sexual reproduction and life cycle of Chara.
sexual reproduction in Chara is an advanced type of oogamy. It is happening during unfavorable conditions like drought, unavailability of nutrients, etc. At the time of sexual reproduction, the members of Chara produces multicellular and macroscopic sex organs. These sex organs are formed at the nodal region of primary laterals or branches of limited growth. In this figure, this is the nodal region of primary lateral and these are the sex organs. And here, this is a male sex organ and is called globule, and this is the female sex organ and is called as nucleus. The members of Chara may be monoecious or dioecious. In monoecious or homothallic members, both globule and nucleus are formed from same plant body. But in dioecious or heterothallic members, there is male and female plants. Male plant produce globule and female plant produce nucleus. Globule is the male sex organ of Chara. It is spherical in shape and is with a yellow to red color at its maturity. In homothallic members of Chara, it is producing just below the nucleus or female reproductive organ. When we analyze the structure of globule, we can see a short pedicel at its base. This pedicel helps for the attachment of globule to the nodal region of primary lateral. The main body of globule is composed of three types of cells. They are outer shield cells, middle manubrial cells, and inner capitulum cells. These are shield cells and are 8 in number. The surface of these shield cells are with infoldings, so they appear as a structure composed of many cells. In globule, these shield cells are arranged in such a way that the structure encloses a cavity in the center. From the inner side of these shield cells, manubrial cells are formed. In this figure, these are manubrial cells and are rod-like structures. At the tip of these manubrial cells, you can see some globose cells. These cells are called primary capitulum. Each primary capitulum produces two or more secondary capitulum. We can see the development of long filamentous structures from the secondary capitulum and are called as antheridial filaments. From each secondary capitulum, 2 to 4 antheridial filaments can be formed, and each of these antheridial filaments are with 100 to 200 cells and are called antheridial cells. These are antheridial cells. From each of these antheridial cells, one anthrozoid or male gametes are formed.
Here we can see the structure of male gamete in Chara. From the figure, it is clear that it is an elongated, spirally coiled and uninucleate structure. The male gamete is provided with two flagella which make them motile. So, the male gamete is an elongated, uninucleate, spirally coiled and biflagellate structure. In each globule, large number of male gametes are produced. When the globule matures, the shield cells get separated and the male gamete or anthrozoid will come out by the dissolution of antheridial cell wall. Nucleus is the female reproductive structure. In homothallic species, they are formed at the nodal region of primary lateral just above the globule. Externally, the nucleus has a pedicel sheath, and corona. It is through this pedicel it attaches to the nodal region of primary lateral. This is the sheath region, which consists of 5 spirally twisted tube cells from the base, and it covers the nucleus except at the apex. At the apex, we can see a crown of 5 cells called corona cells. Internally, the nucleus is with a large egg, below which a stalk cell and a central cell present. In Chara, the fertilization is internal. That means the male and female gamete fused within the female reproductive structure or nucleus. At the time of fertilization, the tube cells of nucleus get separated and a passage is formed for the entry of male gamete into the nucleus. Through that opening, one of the male gamete or antherozooid enter the nucleus
and fuse with the egg, which results in the formation of a diploid zygote. The zygote thus formed secrete a thick cellulosic membrane around them and convert into a resistant spore called oospore. The wall of nucleus also form a cover around this oospore.
This oospore undergo a period of resting stage to overcome unfavorable season. On return of favorable condition, new thallus is produced by the germination of oospore.
Oospore germination happens during the return of favorable environmental conditions. At first, the diploid nucleus migrate to the anterior region of oospore. Then the nucleus undergo meiotic division and forms 4 haploid nuclei. Now a septum is formed between these nuclei and forms an upper uninucleate and lower trinucleate cell. Later, the nuclei of lower trinucleate cell degenerated and the upper cell with a single nuclei undergo one mitotic division and 2 daughter cells are formed. Of these 2 cells, one cell acts as protonemal initial and other cell acts as the rhizoidal initial. From protonemal initial, erect part of thallus are formed, and from rhizoidal initial, lower rhizoidal region are formed. This is the pictorial representation of life cycle of Chara. In Chara, the dominant phase in the life cycle is gametophyte. The diploid phase is represented by only zygote and oospore. So it is a typical example for haplontic life cycle. Gametophytic stage of Chara can multiply by 2 ways. One is by vegetative reproduction and another is by sexual reproduction. There is no asexual reproduction in Chara. Vegetative reproduction in Chara may be by producing bulbils, amylum stars, or by producing secondary protonema. Vegetative reproduction happens during favorable environmental conditions, and in unfavorable environmental conditions, Chara undergo sexual reproduction. For sexual reproduction, they produce multicellular and macroscopic sex organs. Here, the male sex organ is called a globule, which produces numerous male gametes, which are called antherozooids. The female sex organ is called nucleus, which produce single female gamete called egg. At the time of fertilization, both male and female gametes fuse to produce a diploid zygote. Zygote then converted into a thick-walled resistant spore called oospore, which is also diploid. In this oospore form, Chara tide over unfavorable condition. During the return of favorable condition, the diploid nuclei of oospore undergo meiotic division and produce haploid protonymal stage. From this protonema, a new haploid thallus will develop.